Hello students, today we will be studying the structure of dicot and monocot embryo. I am Ms. Lynette Fernandez, Associate Professor, PESS Ravi S. Naik College of Arts and Science, Farmagudi, Ponda, Goa. In this module, we will be studying the structure of dicot embryo, structure of the monocot embryo, developmental stages of dicot and monocot embryos. We will be learning and understanding the structures of dicot and monocot embryos. Let us briefly see what happens during double fertilization. When the two gametes, the male gametes, enter the embryo sac, one male gamete fuses with the egg cell and the other male gamete fuses with the diploid primary endosperm nucleus. The male gamete which fuses with the egg cell forms a diploid zygote or the oospore, and the male gamete which fuses with the secondary nucleus forms a triploid primary endosperm nucleus. Now this zygote which is formed, it ultimately gives rise to the embryo which is present in a mature seed, and this embryo has the potentiality to form a new plant. Let us see what happens during the fertilization or after the fertilization. The zygote in the embryo sac develops into the embryo, and the primary endosperm nucleus develops into the nutritive tissue which is called as the endosperm, which provides nourishment for the developing embryo. The ovule in which the embryo sac is present develops into the seed, and the integuments covering the ovule develop into the seed coat. The ovary develops into the fruit. Now, if you observe the embryo is enclosed. It is a baby plant. It is enclosed in a seed, and it consists of two parts, that is the cotyledon and the embryonal axis. The cotyledon may be papery and a single cotyledon might be present, but in some cases there are two cotyledons present, as in the case of dicots. And the embryonal axis is made up of the plumule and the radicle. Let us see briefly the stages in the development of a typical dicot embryo. The development of the embryo from the zygotic stage is referred to as embryogenesis. The zygote, once it is formed, it goes through a period of rest and it does not divide immediately. In contrast, the primary endosperm nucleus, it divides immediately and does not undergo any period of rest. Once the zygote divides, it divides through a transverse division to form two cells, a basal cell and a terminal cell. The basal cell undergoes the transverse division, and the apical cell, it divides vertically. Thus, it forms a four-celled inverted T-shaped proembryo. Each of the apical cells divide by vertical walls, and this results in a quadrant. The quadrant cells in turn become partitioned by transverse walls so as to form 8 cells, that is an octant, and these are present in two tiers, an upper tier of 4 cells and a lower tier of another 4 cells. So the lower 4 cells of the octant is referred to as the epibasal octant, and these are destined to give rise to the stem tip and the two cotyledons, and the upper 4 cells are referred to as the hypobasal octants, and these give rise to the hypocotyle. All of these 8 cells divide periclinally, forming a globular embryo. Now the outer derivatives of this globular embryo, they ultimately form the dermatogen, while the inner ones, they undergo further divisions and they give rise to the peribleum just beneath the dermatogen, and the plerome, which is in the central region. The cells of the dermatogen later on divide and eventually produce the epidermis of the embryo, and the cells of the periblim, after further divisions, they give rise to the cortex, and the plerome forms the procambial system of the hypocotyle region. Now, meanwhile, the two upper cells which were formed in the proembryo, they divide to form a row of 6 to 10 cells. These are called as the suspensor cells, of which the cell which is closest to the micropyle enlarges, that is the uppermost cell. It enlarges and it swells, it becomes swollen and vesicular, and it probably has a hostorial function. The cells of the distalmost cells of the suspensor, which are contiguous, the distalmost cells of the suspensor which are contiguous with the globular embryo, they develop into the hypophysis, and this hypophysis gives rise to the embryonic root and the root cap. At the same time, in the globular embryo, the outermost cells at 2 points give rise to initials, two initials, and those are the initials for the cotyledons. And in between these two initials, there is another region, another point or another initial, which is called as the plumula initial, which is present between the two or is flanked by the two cotyledonary initials. But the growth in the cotyledonary initials is much faster than that in the plumula initial. So, the globular embryo, as a result of which, develops into a heart-shaped or a chordate shaped embryo. Now, ultimately, the growth takes place in the cotyledonary initials to such an extent that two large cotyledons are formed, and these cotyledons, in order to get themselves accommodated in the embryo sac, which is a curved structure, they also curved, and the entire embryo, it appears to be curved. Let us see the structure of the mature dicot embryo. Now, the embryo is made up of two parts, that is the embryonal axis and the two cotyledons. At the end of the embryonal axis on the other side, there is a swollen suspensor, and the portion of the embryonal axis, this is the embryonal axis, and the portion of the embryonal axis above the level of the cotyledons is called as the epicotyle, and that portion below the level of the cotyledons is referred to as the hypocotyle. Hypocotyle at its further end has a root initial which is called as the radicle, which is covered by a root cap. And the other part of the hypocotyle, or the further end, or the part of the hypocotyle between the radicle and the plumule, develops into the procambial strand. It develops into the procambial strand of the embryo. The epicotyle gives rise to the growing part, which is called as the plumule, and this epicotyle is responsible for giving rise to the entire shoot system. So the epicotyle terminates into the plumule, which is the embryonic shoot, and the lower end of the hypocotyle, it forms the radicle or the embryonic root. In the hypocotyle region, the central cells, they become somewhat elongated and they form the procambial strand. And as I had already said, the pronounced curvature of the cotyledons takes place, and that is due to the enlargement of the cotyledons as well as the enlargement of the cells of the hypocotyle. Now, the monocot embryo In the case of the monocot, the difference between the monocot embryo and the dicot embryo is that in the monocot embryo, there is a single cotyledon present. And in the case of a dicot embryo, there are two cotyledons present. And this monocot embryo in the monocots is present on the lateral side of this embryonal axis. Let us see a few developmental stages in brief of the monocot embryo. The zygote undergoes a transverse division to form two cells, a terminal cell and a large basal cell. The large basal cell simply enlarges and it is transformed into a large vesicular structure which is called as the haustorium, which helps in the absorption of nutrition. Then the terminal cell, it divides by a transverse division to form two cells. The tip cell, which is called as the top cell, and below it is the middle cell. The middle cell is actually responsible for giving rise to almost all parts of the embryo after dividing and redividing. It gives rise to the plumula part, the radicle, the apicotyle and hypocotyle, and also the suspensor. And the top cell undergoes divisions to give rise to the cotyledon, which is a thin papery structure. A single cotyledon is formed by the top cell. So, later on, the suspensor as well as the basal cell, they undergo degeneration. Now, the structure of a mature monocot embryo. Let us see the structure of the mature monocot embryo. The embryo consists of two parts, a single cotyledon and the embryonal axis. In the case of the grass family, this single cotyledon is referred to as the scutellum, and it is present on the lateral side. It is more of a papery type of a structure, very thin and papery type of a structure. And the embryonal axis is made up of the radicle and the plumule. This embryonal axis shows the presence of the radicle on the lower side. So that part of the embryonal axis, which is below the level of this scutellum, is referred to as the radicle. Now the radicle is covered by a root cap. It is covered by the root cap, and this in turn is covered by an undifferentiated type of a tissue which forms a sheath, a protective sheath around the root cap, that is called as the coleorhiza. Then above the level of the scutellum, the embryonal axis, it forms the apicotyle, and this apicotyle is made up of the plumule, which is nothing but the shoot apex, and this is surrounded by the leaf primordia, and these leaf primordia in turn are surrounded by again another protective sheath or a protective covering, which is called as the coleoptile. This coleoptile is made up of undifferentiated tissues. So the entire structure is made up of the scutellum on one side, that is on the lateral side of the embryonic axis. The embryonic axis is made up of the radicle on its lower side, covered by the root cap, and that in turn covered by the protective coleorhiza. And on the opposite side or on the upper side of the level of the scutellum, the embryonic axis shows the presence of the apicotyle, which is the growing shoot, covered by the leaf primordia, which in turn is covered by the coleoptile. On one side of the embryonal axis, there is a small extended structure which might be present in some of the embryos, which is called as the epiblast. This epiblast is probably the remnant of the second cotyledon. Function is not known. Now, if you see the monocot grain, In this case, it's the maize seed. It shows two regions, a larger portion, which is the endosperm, and a smaller portion, which is called as the embryo. Now, this larger portion, or the endosperm, it contains a lot of nutritive reserve food material, and the two are separated from each other by means of an epithelial layer. Now, the nutrition from the endosperm is broken down and it is passed on to the cotyledon, the single cotyledon. From there, it is passed on to the growing embryo for its development. So the difference between the monocot and the dicot embryo is that in the case of the monocots, there's a single cotyledon present, and in the case of the dicots, there are two cotyledons present. In the case of the monocots will show the presence of the cotyledon present on the lateral side of the embryonic axis, and in the case of the dicots, the cotyledons are present on either side of the embryonal axis, and they are almost parallel to the embryonal axis. These are the references. Thank you.